If you bypass the folded outer layers of the human cerebral cortex and travel deep into the absolute center of the skull, you arrive at a structure that physically connects us to the very dawn of vertebrate life. While we often treat the third eye as a spiritual metaphor for intuition, the anatomical record reveals a far more concrete reality. This is a documented evolutionary relic. Deep within the epithalamus, resting near the brainstem, sits the pineal gland. It measures about 5 to 8 millimeters across and, as its name suggests, resembles a tiny biological pine cone. For millions of years, the ancestors of this tiny gland operated on the surface of the head as an external light-sensing organ. Today, it remains a literal physical eye, now trapped in the pitch black of the human cranium. Identifying the pineal gland as a physical organ rather than an abstract concept reveals a hidden layer of our neuroanatomy, a light sensor buried in a region of the body usually dedicated to processing complex data. To find its origin, we have to travel back 518 million years to the Cambrian oceans. Here, we find the Mylocynmingids, tiny, jawless fish equipped with a highly complex, four-eyed visual system. Biologists tracking the fossil record have determined that one specific pair of these ancient camera-type eyes is the direct evolutionary precursor to our modern pineal gland. We can still see this ancient hardware in modern reptiles, like the tuatara, which carries a parietal eye right on the top of its head. This structure isn't decorative. It possesses a functional lens, a cornea, and a retina. Instead of forming complex visual images, it detects the brightness of the sun to help the reptile regulate its body temperature. As the vertebrate lineage evolved toward endothermy, or warm-bloodedness, our mammalian ancestors developed the ability to regulate their internal temperature. A literal skylight on top of the skull was no longer a survival requirement. Slowly, the parietal eye sank beneath the bone, taking up residence deep inside the brain. Yet it retained its ancient photoreceptive genes, known as opsins. The human body is a walking museum. We carry a literal, light-sensing eye, buried alive in the center of our skulls. This presents an immediate biological paradox. A photoreceptive organ requires light to function, but the human pineal gland exists in total darkness. To solve this, the brain uses the pineal gland as a neuroendocrine transducer. When daylight hits our front lateral eyes, the retina sends electrical signals down a multisynaptic pathway, routing the information around the brain and directly into the pineal complex. The gland then acts as a translator. It converts those electrical signals from the eyes into a chemical output, pumping the hormone melatonin directly into the bloodstream. This melatonin release operates on a strict schedule, peaking in darkness and halting in sunlight. It tells the entire body what time it is, regulating our sleep-wake cycles, modulating inflammation, and supporting the immune system. This blind, trapped eye acts as the master conductor of our physiology, dictating our biological time and keeping us alive from the inside out. If this organ is so critical to our internal rhythm, modern neuroscience is now asking a controversial question. Can humans actively control or stimulate it? In early 2025, researchers published a major study in the Journal of Pineal Research, examining the brains of individuals with extensive, long-term meditation experience. This data visualization illustrates their findings. On the left, we see the baseline MRI signal density of a non-meditator's pineal gland. On the right, the scans of the long-term meditators show a significantly denser, brighter, and structurally thicker gland. The biological age of their brains, measured through a score called BrainPad, actually dropped well below their chronological years, indicating a brain that is functionally younger than its birth date would suggest. These clinical observations align with ancient descriptions of Shambhavi Mahamudra, a contemplative practice where the gaze is locked at the ajna center, situated precisely between the eyebrows. We now have the biological measurements showing that these types of focus-based techniques boost brain-derived neurotrophic factor and trigger healthy cortisol responses in the body. Ancient traditions that centered on the third eye appear to have been using highly effective or reverse-engineered biological tools for brain optimization long before we had the technology to verify them. This biological reality gives new weight to the theories of the 17th century philosopher René Descartes, who became obsessed with this tiny organ. Descartes looked at the brain and noticed a pattern. Almost everything comes in pairs: 2 hemispheres, 2 eyes, 2 ears. But the pineal gland is the only unpaired structure, sitting exactly on the midline. To him, it had to be the unifying point of consciousness. He officially dubbed the pineal gland the seat of the soul, the precise physical location where the immaterial mind anchors itself to biology. Fast forward to modern speculative science. Researchers like Dr. Rick Strassman have expanded on Descartes' idea, linking the pineal gland to dimethyltryptamine, or DMT, often referred to as the spirit molecule. Biological evidence confirms that trace amounts of endogenous DMT and the specific enzymes required to synthesize it are physically present inside the human pineal gland. This chart tracks the startling data from a 2019 study on cardiac arrest in rats. As the EKG monitor flatlines, the brain triggers a massive, sudden surge of DMT, reaching concentrations comparable to standard melatonin levels. This raises an incredible question. Could the third eye be the biological hardware that generates the mystical visions people report during near-death experiences? The third eye traveled 500 million years to get here. It transitioned from an external ocean sensor into the internal master clock that dictates our daily biology. Clinical data now confirms that we can actively preserve the structural integrity of this gland, slowing the aging process of the brain through mental focus.